Good morning, everyone. In today's session, I'm going to cover the transport of different substances, including molecules and ions, across the cell membrane or the plasma membrane. We know very well that the plasma membrane separates the internal environment of the cell, which is the cytoplasm, from its external environment, and this is the interstitial fluid for most body cells except the blood cells. The objectives are as follows. The first objective is to describe the structure of biological membranes, and here we mean the cell membrane, and the functions of the different plasma membrane proteins. The second objective is to discuss the transport mechanisms of molecules and ions across the cell membrane, and finally understand the concepts of osmosis, osmolarity, osmotic pressure, and tonicity. The cell membrane is composed of phospholipid bilayer, as we can see here in this slide. This is a segment of the cell membrane, and it is composed of phospholipid bilayer plus cholesterol that is dispersed here and there, and not to forget one of the most important parts of the cell membrane, and these are the membrane proteins. If they are fully spanning the membrane protein, we call them if they are fully spanning the phospholipid bilayer, we call them integral membrane proteins, and if they are attached to the surface, we call them peripheral membrane proteins. So, according to the previous slide, we have at least two different types of membrane proteins. Those that fully span the membrane, the phospholipid bilayer, we call them integral membrane proteins. These are transmembrane receptors, ion channels, and carrier proteins, and we'll cover them in the coming slides of this session. And we have some proteins that are not spanning the entire thickness of the phospholipid bilayer, and we call them the peripheral proteins, like the ankyrenes in the red blood cells. Now, what are the functions of the different membrane proteins? We should know at least 6 different functions of the membrane proteins. Number 1 is transport. Many membrane proteins act as transporters of different molecules and ions between the exterior and the interior of the cell. Some molecules or ions can cross the cell membrane from the exterior to the interior, and others can cross in the opposite direction. Some transport mechanisms are passive, they don't need energy, so they don't require ATP. Others are active and they require ATP, and we'll see examples at the end of this session. Another important function of membrane proteins, we know that enzymes are proteins, so some of the membrane proteins have enzymatic activity, where they can convert a substrate into products, and then another membrane protein can deal with the product of the first reaction as substrate to produce another product. And in physiology, we have many examples about the enzymatic activity of membrane proteins. Another important function for membrane proteins is their ability to act as receptors for signal transduction. And the best example I can give is are the receptors for acetylcholine, one of the most commonly used neurotransmitters by our nervous system. And acetylcholine is the neurotransmitter. And in order to produce its effect on the cells or the target cells, it should bind to specific receptors. We call them the cholinergic receptors. Other functions of the membrane proteins are the intercellular adhesion. So we have many examples where cells can adhere to each other using membrane proteins. We have the tight junctions that are very common. Also, we have the gap junctions that connect the cytoplasm of two adjacent cells. And in physiology, there are many examples about tight junctions, like in the epithelium of many tissues, and the gap junctions like the connections between the cardiomyocytes and the heart. Another important function of the membrane proteins is their ability to or allow cells to recognize each other. And the best example for this is the cell-cell recognition that happens during an immune response. Another function, which is the last function here in this slide, is the membrane proteins might act as a site for the attachment of the cytoskeletal proteins and the extracellular matrix proteins. So they are very important. As I said previously in one of the sessions, they can maintain the shape of the cell, they help in the motility of the cell. In some types of the cells like muscles, they help in the contraction, and also they help in the attachment of the cells to their neighboring cells and the extracellular matrix. Now, how different substances like molecules or ions or even sometimes large solid particles can cross the cell membrane, which is mainly composed of phospholipid bilayer. As we can see, some substances based on their chemical nature, they are lipid soluble, so we call them hydrophobic. They can cross the phospholipid bilayer directly by passing through the phospholipid bilayer, or if they are lipid insoluble, they need a channel protein to cross the phospholipid bilayer. This type of transport is called simple diffusion, and the driving force here is the concentration gradient. If I have the concentration of the molecule high on one side and low on the other side, these molecules will move from the high concentration to low concentration across the cell membrane, either directly or through the channel protein. This is called simple diffusion. In other cases, we have large molecules that need to bind to a certain membrane protein called the carrier, and once they bind to the interior of that carrier, they will cause change in the shape of the protein, conformational change in the protein. So the carrier will open on one side and release that molecule to that side. So this is called facilitated diffusion, and this type of transport is again down the concentration gradient from high concentration to low concentration. Both simple diffusion and facilitated diffusion are under the umbrella of passive transport. Why we call it passive transport? Because here we don't need ATP as a source of energy. The driving force is just the concentration gradient, because on one side we have high concentration of the molecule or the ion, and on the other side we have low concentration, and this concentration gradient will make sure that that molecule will be transported from the high concentration to the low concentration across the cell membrane. Another type of transport called the active transport. It is called active because we need ATP. We need ATP either directly, and this is called primary active transport, or indirectly, and this is called secondary active transport. And I'll explain that in details in the coming slides, the main differences between primary and secondary active transports. Now let's start with the different types of transport across the cell membrane, or let's summarize here the different types of transport across the cell membrane and then discuss one by one in more details. So when we talk about transport across the cell membrane, we have the passive transport. It includes simple diffusion. It happens through the cell membrane, directly through the phospholipid bilayer, or through channel protein. And the best example for channel proteins are the ion channels. Channels for sodium, calcium, chloride, and other ions. Then we have osmosis, and this is the term that we use specifically to describe the transport of water across the cell membranes. So whenever we hear the word osmosis, it means the transport of water molecules across the cell membrane. Then we have facilitated diffusion for larger molecules, and we need here a carrier protein. And there is another way of transport called the filtration. And this type of transport is driven by hydrostatic pressure difference. Hydrostatic pressure caused by the presence of fluid. And this will be covered more in the cardiovascular system when we are going to talk about the transport across the capillary wall, and also to some extent in the renal system when we are going to talk about glomerular filtration. The other entity of transport across cell membrane is active transport, and it is divided into primary active transport, secondary active transport, and as I said here, we need ATP as a source of energy, and whether we use ATP directly or indirectly, we subdivide active transport into primary and secondary transport. Then we have another type of transport called the vesicular or the bulk transport. For larger molecules or for large solid material that cannot cross the cell membrane by passive and active transport, we use the vesicular transport or the bulk transport. And this includes phagocytosis, endocytosis, and exocytosis. So phagocytosis, endocytosis from the exterior of the cell into the cell, materials are taken into the cell inside vesicles, or exocytosis, materials are released from the cell to the exterior via vesicles, and this is called exocytosis. Now let's start with the passive membrane transport. Number one, simple diffusion, and this happens either directly through the phospholipid bilayer or through channel proteins. And the best example for channel proteins are the ion channels. It doesn't need any ATP, that's why we call it passive transport. The different substances diffuse either directly through the phospholipid bilayer or through channel proteins, and as I said, ion channels are the best example. Here we transport lipid-soluble substances and ions. So any lipid-soluble substance can cross directly the lipid bilayer, this phospholipid bilayer. Ions that cannot cross the phospholipid bilayer, they need these channel proteins, and we call them ion channels. Now, what are the factors that affect simple diffusion? We have several factors. These include the concentration gradient, the size of the solute or the molecules or ions, the surface area of the membrane, the thickness of the membrane, the temperature, and another factor. So these are the concentration gradient. The higher the difference in concentration, the higher rate of simple diffusion. Electrical gradient, and this applies only for ions, for charged atoms, the ions. So when we have positively charged ions on one side and we have negativity on the other side of the membrane, this will attract the ions, so we have a faster rate of diffusion. As I said, surface area for diffusion, if we increase the surface area of the membrane, this will increase the rate of simple diffusion. The membrane thickness, the thinner the membrane, we have the higher rate of transport. If the membrane becomes thick, this will hinder or will decrease the rate of transport. Then we have the mass or the size of the diffusing molecule or ion. The smaller the size of the ions or molecules, they diffuse faster. And finally, the temperature of the solution. With higher temperature, molecules have higher kinetic energy, so they can diffuse at higher rate. Now let's talk about transport through protein channels. And here the best example, as I said, the ion channels. Ions are charged, therefore they cannot cross the phospholipid bilayer, so they cannot directly cross the phospholipid bilayer. That's why they need ion channels. Ions move in and out of the cell through ion channels, and here we can see one of the ion channels. It has a pore that contains water, and this pore that contains water is the path through which ions can enter or leave the cell. Ion channels are transmembrane proteins that transport ions in and out of the cell. They transport ions down their chemical gradient, also we call the concentration gradient, and because ions are also charged, we should mention the electrical gradient. So ions move across the membrane through ion channels down their electrochemical gradient. There are two gradients that affect the movement of ions, and of course we don't need ATP. That's why transport through ion channels is one of the passive transport mechanism across the cell membrane. Ion channels have two important features or properties. Number one, selectivity, so they allow some ions to pass but not others, and we'll see examples in other sessions. We have potassium channels, they are very selective for potassium. We have the sodium channels, we have the calcium channels, and the chloride channels as examples. Of course, at higher level you recognize that we have some ion channels that are non-selective, but they preferably transfer one or transport one of the ions. For example, we can say non-selective cation channel that transports positively charged ions, but they are more permeable to calcium, for example. Another important feature of the ion channels is gating. Ion channels open and close in response to specific stimuli, and based on the stimuli or the stimulus that opens or closes the gate of ion channels, we divide ion channels into different types. Here you can see a type of ion channels. This is a potassium channel. These channels, their gates open and close randomly without any stimulation. That's why we call them the leak channels. And potassium leak channels are the most abundant leak channels that are present in our cell membranes. Then, if the channel gate opens or closes in response to the binding of a chemical stimulus or molecule like acetylcholine, then we call them ligand-gated ion channels. Also, we can call them chemically gated ion channels. And here, for example, the acetylcholine receptor. We said this is one of the famous neurotransmitters used in our human body. If the gates open or close in response to mechanical stimuli, we can call them mechanically gated ion channels. And every single cell in our body is exposed to mechanical stimuli. So we have abundance of mechanically gated ion channels in our different body cells. And then we have another type of the gated ion channels that open or close in response to the change in membrane potential or in membrane voltage. For example, this channel at membrane voltage or membrane potential of minus 70 millivolt, the gate is closed, but if the membrane potential becomes minus 50, the gate will open and this will allow ions to move. And since here this is a channel for potassium, potassium will move across the cell membrane through this voltage gated potassium channel. Of course, not to forget that our body is exposed to different temperatures, from cold to hot. That's why we have another type of gated ion channels. We call them temperature regulated or thermally gated ion channels. So we can add these channels to the above or to the channels that we mentioned here in this slide. So the gating, which is opening and closure of ion channels, occurs in response to specific stimuli. At least we have four types. We have leak, non-gated channels are always open. These are potassium leak channels, and also we have sodium leak channels. But potassium leak channels are the most abundant in our cells. Then we have the ligand-gated channels that open and close in response to extracellular mediators like acetylcholine or other inhibitory neurotransmitters. We have ligand-gated ion channels that respond to intracellular ligand like calcium and ATP. And in physiology we'll encounter many examples. Then we have the mechanically gated ion channels that respond to mechanical vibration or pressure. So these are all mechanical stimuli. We have the voltage-gated channels that respond to direct change in the membrane potential or change in the voltage of the membrane. And we have voltage-gated sodium, voltage-gated potassium, and voltage-gated calcium channels. And these are very important for the action potential in neurons and muscles, while voltage-gated calcium channels are very important in the synapses. They are very important for the exocytosis of neurotransmitters or even the hormones. The voltage-gated ion channels are responsible for the generation of action potential, especially the voltage-gated sodium and potassium channels. As I said, we don't forget the temperature-regulated or temperature-gated are also called thermally gated ion channels that respond to changes in temperature. Let's move to the second type of passive membrane transport, which is osmosis. Whenever we read or hear the word osmosis, it is the transport of water, only water across the cell membrane. So this term is just to describe the transport of water across the selectively permeable membrane, and in the biological system we are talking about the cell membrane or the plasma membrane. So osmosis is the net diffusion of water across a selectively permeable membrane. Water can pass through the plasma membrane in two possible ways or in two different ways. As you can see here in this figure, water is a small non-polar molecule, so it can pass directly through the phospholipid bilayer, or it can pass through special channels that are selective for water, and these are channel proteins present in the cell membrane. They span the full thickness of the phospholipid bilayer, and they provide a pore for the water movement between both sides of the cell membrane. So water can move through these channels, and we call these channels aquaporins. It is a large family of water channels, and as I said, they are integral membrane proteins. Let's know about osmolarity and how it is different from osmolality and tonicity. Whenever we mention osmosis, which is the diffusion of water across the cell membrane, we should also know what is osmolarity, what is osmolality with an L, and tonicity. Osmolarity is the measure of solute concentration per unit volume in liter of the solvent, and here the solvent is water. This is the universal solvent in all biological systems, and the presence of solutes in the solvent, which is water, will create an osmotic pressure. If we have any solute in a solution, this will create an osmotic pressure. Osmolality with an L is the measure of solute concentration per unit mass or kilogram of the solvent. So when we dissolve solute in 1 kilogram of water, then we will get the osmolarity. The concentration of that solute in 1 kilogram of water will generate an osmolarity, and this will lead also to the presence of osmotic pressure in that solution. Tonicity: if I have two solutions that have different osmolarities, the osmolarity gradient or the osmotic pressure gradient between both solutions that are separated by a membrane is called tonicity. So tonicity is the measure of osmotic pressure gradient between two solutions that are separated by a semi-permeable membrane. And as I said, the best example of semi-permeable membranes in any biological system is the cell membrane or the plasma membrane. Unlike osmolarity, tonicity is only affected by the solutes that cannot cross the semi-permeable membrane, because these are the only solutes that affect the osmotic pressure gradient. So any solute that is present in any solution on both sides of the cell membrane that cannot cross the cell membrane are those contributing to the tonicity or the osmotic pressure gradient. Osmolarity is proportional to the number of osmotic particles formed per molecule once the molecule dissolves. We know very well that some molecules, when they dissolve in water, they dissociate into smaller particles or into ions. For example, sodium chloride molecule, when it is dissolved in water, each molecule will give me 2 ions. sodium ion and chloride ion. And each one of these ions is considered as a solute in the solution and will play a role in the osmotic pressure. So osmolarity, which is in osmoles per liter, is the number of moles multiplied by the number of particles that are generated per molecule in the solution. For example, if I have 1 mole of sodium chloride dissolved in water, this will give me 2 osmolar solution. A solution that has an osmolarity of 2 osmoles per liter, because as I said, 1 molecule of sodium chloride powder in the solution will give me 2 ions. Another good example, if I have 1 mole of calcium chloride that will be dissolved in water, this will give me, in 1 liter of water, will give me a solution that has an osmolarity of 3 osmoles per liter. Why? Because 1 molecule will give me 3 ions. 1 calcium ion and 2 chloride ions. So in the solution I have triple the osmolarity. Physiological concentrations, of course, the concentrations of solutes in biological systems or in body fluids are very low. So our body fluids are very diluted, so it is not proper to use the unit osmoles per liter. We use the unit milliosmoles per liter. So milliosmol is the unit that is most appropriate to describe the concentration of solutes in our body fluids. And 1 milliosmol per liter is equal to 10 to the power minus 3 osmol per liter. And since our body fluids are very diluted, the osmolarity and osmolality are almost equal. Very small difference between them, and I usually prefer to use osmolarity across the board in all physiological sessions. Normal values for our body fluids is between 290 to 300 milliosmoles per liter. So this is the normal osmolarity of our body fluids, either inside the cell or outside the cell. Any change in osmolarity that exceeds 2%, any decrease or increase in this osmolarity in terms of 2% will start physiological processes that will try to bring back the osmolarity to the normal range. Osmolarity increases either by decreasing the water, the volume of the solvent, or by increasing the amount of the solute, and this is quite logical. Normally, cells neither shrink nor swell because the intracellular and extracellular fluids have the same osmolarity. So the fluid that is present in the cells, the cytoplasm, has the same osmolarity of the extracellular fluid, so there is no change in the volume of the cells. They will not swell nor shrink. If we want to change the volume of the cell, then we have to play with the osmolarity of the fluid outside the cell. Now let's calculate the osmolarity of important solutions that are used in the hospital. One of the common IV solutions or intravenous solutions that are used in hospitals is 0.9% weight per volume sodium chloride solution. What do we mean by 0.9%? 0.9% means that we dissolve 0.9 grams of sodium chloride in 100 ml of distilled water. Or it means that we dissolve 9 grams of sodium chloride in 1000 ml of distilled water. So 9 grams of sodium chloride powder, and we know very well that the molecular weight of sodium chloride is 58.5. It is 23 for sodium and 35.5 for chloride, so the total or the molecular weight of sodium chloride molecule is 58.5. How many millimoles are present in 9 grams of sodium chloride? We divide 9 grams over 58.5, and this will give me 0.14 mole or 154 millimole. So we are talking about millimole. If we put 154 millimole of sodium chloride powder into 1 liter of water, this will give me a solution that has an osmolarity of 308 milliosmoles per liter. Why? you can notice that the value here 308 is just 154 multiplied by 2. Why? Because each molecule of sodium chloride will dissociate in the water and give me two osmotically active ions, the sodium and the chloride. See, so the osmolarity here is 308, which is very close to the osmolarity of our body fluids. That's why the 0.9% sodium chloride solution is iso-osmolar. It has almost the same osmolarity of our body fluids. Now, what is the osmolarity of 5% weight per volume of glucose solution? What does it mean by 5% glucose? It means that we dissolve 50 grams of glucose in 1000 ml of water, or 1 liter of distilled water. So we have 50 grams of glucose, and we know very well that the molecular weight of glucose is 180. So how many moles are present in 50 grams of glucose? Just we divide 50 over 180, and it is about 0.278 moles, or 278 millimoles. If I add 278 millimoles of glucose to 1 liter of distilled water, I will get a solution that has an osmolarity of 278 milliosmoles per liter. Why it is the same? Because glucose does not dissociate. The glucose molecule will remain as one molecule when it is dissolved in water. It won't give me more than one molecule. So the molarity and osmolarity are similar. Now, what happens if I use, or if I put cells in solutions of osmolarities that are different than the osmolarity of the cell? We know very well that the osmolarity, or the normal range of osmolarity of a cell, is between 290 to 300 milliosmoles per liter. We know very well that the exchange of water between the intracellular and extracellular fluids occurs by osmosis, which is the simple diffusion of water across the cell membrane, and this is driven by the osmotic pressure gradient, and we said that the osmotic pressure gradient is the concept of tonicity. If we place the cell in a solution that has the same osmolarity of the cytoplasm, this solution is called isotonic. So solution that has similar tonicity as the intracellular fluid, and here we can use 0.9% sodium chloride. So what happens? Actually, the cell will not be affected because the number of water molecules entering the cell is equal to the number of water molecules leaving the cell. So there is zero net movement of water, no net movement of water. What happens if I put the cell in hypertonic solution, like seawater that has very high concentration of the solute? The solution has an osmolarity more than 280 or more than 300. Now the high osmolarity of the solution will drag water from inside the cell to the outside of the cell. Removing water from the cell will cause the shrinkage of the cell. So the cell will shrink. Shrinkage of the cell means that the cell will lose its function. What happens if I place the cell in hypotonic solution, or even distilled water that has zero solutes inside that? Hypotonic solution, so the water will move from the solution into the cell, because now the cell has an osmolarity higher than the hypotonic solution. Water will enter the cell, gradually the cell will swell, and if this does not stop, the cell will rupture. This will lead to the rupture of the cell. Of course, water moves in and out very fast, so we can reach osmotic equilibrium very rapidly between the intracellular and extracellular fluids. Another form of passive membrane transport, after finishing the simple diffusion, the osmosis, now we come to the third type, which is facilitated diffusion. And this facilitated diffusion, as you can see here, it needs a carrier protein. The transported molecule should bind to a specific point inside the carrier protein. Once it binds to that site, the shape of the protein will change, so it will open on the opposite side to release the transported molecule. So this facilitated or this type of passive membrane transport requires a carrier protein, which is a membrane protein or transmembrane protein or an integral membrane protein. It transports larger molecules such as simple sugars, such as glucose. And the best example are the glucose transporters. We call them glucose transporters. This is a big family. We have GLUT1, 2, 3, 4, and you will encounter that in many occasions. Now, any transport that uses a carrier has some kind of kinetics, certain kinetics. Kinetics of carrier-mediated transport, it includes facilitated diffusion that I covered in the previous slide, and also it includes the primary active transport as well as the secondary active transport that we will cover later on in this session. The carrier-mediated transport shares the following features or the following characteristics: specificity, competition, and saturation. So carriers, we have carriers that are specific for glucose, another carrier specific for another molecule. Competition, some carriers, drug or a molecule competes for the transport of another molecule. And finally, the saturation. And this saturation is because we have a limited number of carriers in the membrane, and once all the carriers are occupied, we cannot increase the transport rate. As you can see here, the carrier-mediated transport, as we increase the concentration, the transport rate will increase until we reach a transport maximum, where all the carriers that are present in the cell membrane are occupied by or are busy with the transport of that molecule. The number of carriers available in the plasma membrane places an upper limit called the transport maximum on the rate at which carrier-mediated transport can occur. And this is very true. For example, why in diabetic patients glucose appears in the urine? Because the large amount of glucose that is filtered in the glomeruli, we have transporters in the renal tubule, especially in the proximal convolute tubule, that will transport glucose back to the blood, but these transporters have transport maximum, and if we reach that transport maximum, any excessive amount of glucose will be passed into urine, and this is one of the main signs of diabetes, which is glucosuria. The fourth type of passive membrane transport is filtration, and this is the passage of water and solutes as a bulk, so water and whatever present in that water through membranes, and this is driven by hydrostatic pressure. For example, the hydrostatic pressure inside the blood vessels will force fluid or water with all its contents that can cross the capillary membrane to pass from the lumen of the capillary into the interstitium. The pressure gradient pushes solute containing fluid from high pressure area to lower pressure area. Now let's move to the active transport. And the active transport requires two things. Number one, it requires a carrier protein. And the second important requirement is the ATP. So these transport systems need ATP to drive the transport because it is transport against the gradient. These carrier proteins use energy to transport ions and molecules from area of low concentration to area of high concentration, something we call against the gradient, against the chemical gradient or against the electrochemical gradient if it is an ion. So the active transport system transports or uses ATP to move solutes across a membrane directly or indirectly. We have two sources of cellular energy can be used to drive the active transport. In the primary active transport, energy is obtained from the direct hydrolysis of ATP by the carrier protein. In the secondary active transport, the energy is obtained from the ionic concentration gradient that was created by the primary active transport. Which means that if the primary active transport fails, the secondary active transport will fail. So secondary active transport depends on the primary active transport. Without primary active transport, all the secondary active transport systems will fail. So based on the previous explanation, we have the primary active transport, and the best example in our body is sodium-potassium ATPase pump. This pump is present as an integral membrane protein as part of the cell membrane. This pump transports 3 sodium ions from inside the cell to the outside in exchange for 2 potassium ions from outside to the inside of the cell. And for this transport, we need 1 ATP molecule. So for each ATP, 3 sodium are extruded from the cell and 2 potassium are brought into the cell. As you can see here, ATP is directly used by the carrier, which has ATPase activity, and this is primary active transport. The primary active transport, by pumping sodium outside the cell, will create a concentration gradient for sodium. So sodium will be very high outside and very low inside the cell. So the concentration of gradient for sodium will be used by the secondary active transport to transport another molecule against its gradient. Here we have the secondary active transport. You can see that no ATP is directly used. The pump or the carrier is using the sodium concentration gradient that was created by the primary active transport to co-transport or transport glucose at the same time. Glucose is transported from low concentration outside the cell to high concentration inside the cell. And here this is sodium glucose co-transporter. We have many examples of secondary active transport, and we'll see a lot of them in different topics in physiology. So types of active transport or membrane transport, we divide them according to the way of ATP usage as primary active transport, where hydrolysis of ATP will phosphorylate the transport protein, causing conformational change, and this will allow the movement of ions against their concentration gradient. The secondary active transport, it uses the exchange pump uses the gradient that was created by the primary active transport to drive the transport of another solute. And the most, or sodium, is the most important gradient that is used by many of the secondary active transport systems. And this sodium gradient is generated by the primary active transport or the sodium-potassium pump. We can also classify the active transport according to the direction of solute or substance movement, because we saw in the active transport usually 2 or more ions or molecules are transported at the same time. So if the 2 substances are moving across the cell membrane in the same direction, we call this symport or co-transport. And in the previous slide, we saw the sodium-glucose co-transport system. If the substances are moved across the membrane in opposite directions, we call that antiport or counter-transport system. And the sodium-potassium pump is a good example of the antiport or the counter-transport. So as you can see here, we have symporters like the sodium-glucose symporter or co-transporter, and we have the sodium amino acid symporter. You can see that the gradient, sodium gradient that was generated by the primary active transport is driving those secondary active transport systems. Here we can see on the left side of the slide, we have the transport of hydrogen from the interior of the cell to the exterior, while sodium is transported down its gradient. These 2 ions are moved or transported against each other. So this is an antiport system or counter-transport system. The same applies for this antiporter, which is the sodium-calcium antiporter. The last type of transport across the cell membrane is the vesicular transport, or called the bulk transport. And this is for the transport of large particles and macromolecules across the plasma membrane. We have phagocytosis, like solid particles, bacteria, fragments of dead cells or dead cells, whatever, are engulfed into the cell inside vesicles that we call them phagosomes. Also, the cell can take a small amount of extracellular fluid into the cell, and this is a process called pinocytosis. So the cell is drinking fluid from outside the cell into the cell, and this process is non-specific. The phagocytosis and pinocytosis are non-specific, but if the process is mediated by receptors present on the cell membrane, then it becomes specific, and we call receptor-mediated endocytosis. So in vesicular transport, we have different types. Of course, cell can take things into the cell and also can release products from the cell by a process opposite to the endocytosis and phagocytosis. So we have phagocytosis, where pseudopods engulf solids and bring them into the cell's interior. We have endocytosis that enables large particles and macromolecules to enter the cell. And we have the opposite to phagocytosis and endocytosis, which is exocytosis, movement of substances from the cell interior to the extracellular space. And the best example is the release of neurotransmitters from nerve endings, where vesicles containing, for example, acetylcholine are releasing their content of neurotransmitter molecules in the synaptic cleft at the synapse. So phagocytosis, phagocytic cells use pseudopods to surround and engulf particles. The pseudopods will join on both sides, fuse together and surround the ingested particle. This is called phagosome or food vacuole. Then lysosomes digest the food vacuole, and this process protects from invading organisms like bacteria and also removes debris and dead cells from our body. There's something called bulk phase endocytosis or pinocytosis, which is drinking by the cell. This is a non-specific process because it doesn't involve any receptors. The plasma membrane invaginates, fuses, and the vesicle containing extracellular fluid pinches off, and the vesicle enters the cell. If the process is specific and mediated by receptors, it is receptor-mediated endocytosis. There is an interaction of the molecules in the extracellular fluid with the specific membrane receptor proteins. The membrane invaginates, fuses, pinches off, and forms a vesicle. The vesicle will enter the cell. It is similar to the previous process, except that this one is more specific because the molecules in the extracellular fluid will bind to receptors on the cell membrane to trigger the endocytosis. Exocytosis, of course, is a process by which cellular products are secreted into the extracellular environment. Proteins and other molecules to be secreted are packaged in vesicles by the Golgi apparatus. Then the vesicles will fuse with the plasma membrane and release their contents in the extracellular environment. And here calcium ion plays very important role in the process of exocytosis. Thank you very much for following this session. I hope that you got most of the concepts and wish you all the best.